My name is Oliver Schnakenberg. I'm the Deputy Consul General, and it's a great pleasure to open this discussion co-organized by the German Center for Research and Innovation and the University of Cologne. A first co-production tonight, and hopefully many more will follow. May I just take this opportunity to say a few words about the German House. Just as the skin is the interface between the self and the world, the foreign missions are the interface between the German government and the public abroad. I worked hard on this comparison, as you can imagine. So we have the two main institutions in this house are the permanent mission with the United Nations and the German consulate. The permanent mission represents Germany with the United Nations and all its organs and sub-organs, with the Security Council maybe being the most important one lately. And we have some more than 50 people from Germany working in the mission. And the Consulate General is, let's say, the smaller brother. My organization is sort of trying to translate our foreign policy to the American public. So what we are doing at the United Nations Security Council, what we are doing with the Euro or with renewable energies, all this we try to communicate and to promote vis-a-vis the US in this tri-state area. But then we have a number of very interesting other tenants in this house. And I would like to mention the German Research Foundation, represented here tonight by Eva Maria Streyer, and also the German Academic Exchange Service. Both organizations are, by the way, have the leadership in a third and also very important organization that is co-organizing this event, and this is the GCRI, the German Center for Research and Innovation. The center has been established in February 2010 with the objective of promoting Germany as a location for research and innovation. And I can say that it's been quite successful in this respect. The center has been organizing lots of events like this one, and is also a great partner of the consulate, again, in our activities to promote Germany. And besides, and to complete or maybe to also improve the synergies in this house, we accommodate 8 liaison offices of German universities. And among them, the University of Cologne is the latest, and I would like to welcome and thank Stephanie Grupp, who is representing University of Cologne here since last year, and thank you for also organizing this first event, and that will be followed hopefully by many others. So without further ado, I'd like to conclude at this point, and I would now like to invite David Bickers to continue. Thank you very much.
Good evening, ladies and gentlemen. It's a pleasure for me to be here with you, and to I think my job is to act as facilitator and introducer. We want this to be an exchange. It's not just people talking at you. So at the conclusion of each of our 2 presentations, I would certainly urge you to ask questions and engage in dialogue. So what I thought I would do briefly is give you a little introduction to the history of German-American dermatology. And it's actually a quite interesting history. But first, I just want to remind you that you're here, in case you've forgotten, to hear something and learn something about the skin. And the skin is the largest organ of the body. It is, as was pointed out already, a major interface between the body and the environment. It's an incredibly complicated structure that serves a variety of crucial biological functions including barrier function, immunologic functions, neurologic functions, etc. And you're going to learn and hear about those with our two speakers this evening. So German American dermatology has in some ways gone through two historical phases. In the early part of the history of this country, there was no formal medical education, anything like what was already established in Europe. And so in the 19th century, if you wanted to study skin disease in this country, you really had to go abroad. And there were major centers in London, Paris, Vienna, in numerous cities in Germany where people went and studied and learned about skin disease, skin biology, and then came back here and typically opened private practices to see patients. Some practitioners did some teaching in medical schools, but dermatology was very much a very small sort of cottage industry in its early days. Then with the changes that took place in Germany in the 1930s, we had a reverse migration, and sizable numbers of dermatologists and other physicians and scientists came to America, and many came to New York. And dermatology, as a result of this and some other factors, began to grow and flourish in this country. I'm not going to go through details on this, but I'm just trying to illustrate the fact that there have been multiple departments of dermatology in Germany and in America have had fundamental cooperation and historically have enriched one another. And dermatology today represents a truly global specialty with outstanding contributions and contributors in countries all over the world, but in particular, I would say, in Germany and in the United States. In my own department at Columbia, we have a long tradition of having welcomed German American dermatologists. Here you see Willie Kurth and his wife Helen Ollendorf Kurth, who were on our teaching faculty for over 40 years. They were in private practice in northern Manhattan, but worked in our clinic without compromise and were great and wonderful teachers. And Helen Kurth made some fundamental contributions to our understanding of how the skin can be if you will, a beacon or a sign of internal disease. She was one of the first people to recognize that a skin condition was actually a manifestation of an internal cancer, and that if you recognize the skin sign early enough, you might discover the cancer early enough to treat it. The condition was called acanthosis nigricans, and it was an indicator of gastrointestinal cancer. Another distinguished German-American dermatologist was Gerd Steigleder. And Gerd came to Columbia for 2 years in the late 50s, early 60s when Carl Nelson was the professor at Columbia. And in fact, almost decided to stay in America, but returned to Germany and ultimately became chairman of the department in Cologne, and in fact was the predecessor of Professor Krieg, who is currently the chair in that department, from whom you'll be hearing shortly. I would call your attention to the fact that a major advance in our understanding of a rare disease was made at Columbia by John Menkes and Dr. Steigleder, whose name is on this publication in 1962, which was the first recognition of a disease, a complicated disease with fundamental changes in hair, and you're going to hear about hair from Dr. Cristiano, so I had to pull something in with hair. This has come to be known as Menkes hair disease or Menkes kinky hair disease because of the structural aberrations that occur in the hair associated in this case with abnormalities in copper metabolism. Here you see a picture of these findings of the hair in these patients who suffer from multisystem disease and often die early in childhood of neurological complications. And finally, I just want to emphasize that in the past, there have been organizations dedicated to fostering ties between German and American dermatology. And I was fortunate enough to be a resident under the chairmanship of Professor Rudolf Baer at NYU, who is shown here. And he, too, emigrated to this country in the 1930s and ultimately became the chairman at New York University. And we established an organization that we call the Bicontinental Association for Education and Research, or BAER. And this is a group that was at a meeting in 1993. And I'd call your attention to this distinguished gentleman here, who was with us on that occasion and is with us tonight. It's my great pleasure to introduce to you Professor Thomas Krieg from the University of Cologne. And he is going to address the topic, one of the topics of his research interest, which is that of wound healing. Thomas?
Thank you very much, David, for the introduction. And I completely forgot this picture, I must say, which is already now a number of years ago. It's really a great pleasure for me to be here today and to visit the department and also to discuss with my friends and colleague from Colombia two diseases for tonight, Alopecia Areata and chronic wounds, which both represent a severe burden not only to the patient, but also, I think, for the society. Both have in common that they are very frequent diseases, but both target different age groups. However, before going into detail, talking about chronic wounds, I would like to bring you up with a little bit of information and give you some greetings from our University of Cologne, which is actually hosting our Department for Dermatology. It's also hosting the Cologne Cluster of Excellence, funded by the German Research Organization, and it's also hosting our Center for Molecular Medicine. So, let's see how it works. So, University of Cologne is actually a very classical full university in Germany. It is an old university. It was founded 1388, so it's one of the oldest in Germany. However, it lasted only a few hundred years, and then Napoleon invaded the Rhineland, and he brought several things with him. He brought with him some French lifestyle, which we still like in Cologne and still have in Cologne, but he also brought in some laws. And he wanted to bring in laws for the professors, and Cologne professors do not really want to abide laws, they like their freedom, and they had some conflicts then, and what Napoleon did was just closing the university. This did last for actually a number of years, and then the University of Cologne was reopened by the citizens of Cologne, and there's still a very nice contact between Cologne and the citizens, much better contact sometimes than between the university and the ministry in Düsseldorf. So it was reopened, and now Cologne is actually one of the largest universities in Germany. It has 38,000 students, 6 faculties, more or less covers beside the technical areas all the different subsets. During the last 10 years, Cologne University has very much succeeded in concentrating on a limited number of high-profile research areas which have international visibility. And besides several of those, I'm happy to tell you that life science is one of those high-profile research areas, actually. And this is a cooperation between the science faculty and the medical faculty, which we have been trying to encourage during the last years, and we have a very nice joint appointments between both faculties and really have cross-border between all the different disciplines there. The medical faculty has then concentrated very much in trying to understand the molecular basis of human diseases and has joined forces with two Max Planck institutes, which we have of our campus, one for neurological research and one for aging-associated diseases, and also with two Helmholtz centers, one in Jülich, which is concentrating on neurological research also, and another one which has been really recently established between Cologne and Bonn and Jülich, which is located in Bonn and which is concentrating on degenerative neurological diseases. So this cooperation between the Max Planck Institute, Helmholtz Institution, and the university has led to a number of international interactions all over the world, including the United States, including New York, and also the starting of the interaction of the skin programs of Columbia and in Cologne are part of this of these interactions. Actually, the topic of our research, which I would like to introduce to you today, chronic wounds, is also coming from an interaction between the different faculties and between the Max Planck Institute and the university in Cologne, and also with the skin programs here in Columbia. I will talk about chronic wounds, and this is actually the work of an independent research group which we have in our department, which is headed by Sabine Eming and a number of co-workers, and more or less most of the experiments which I try to discuss with you today have been done by those young people. So I would like to start with the case report, with the story of a 70-year-old patient who was in very good health without any cardiac or internal problems. He had a very active life, was working as a lawyer, uninterrupted. However, he was always working seated, in a seated activity at the desk. He did had chronic venous diseases, like you see here, already since several years, but he didn't care very much about that. Then suddenly itching occurred, patient developed swelling of the lower leg, and then a very small wound developed. He didn't care very much about that either. Then suddenly severe pain, redness, and fever was noticed. Patient was hospitalized, and it was quite clear that he did have an acute infection from this small wound starting. So acute infection was successfully treated in a short period of time. Patient was dismissed from the hospital. However, this infection led to an enlargement of the ulceration with no tendency to heal anymore, in spite of a number of local topical therapies. The pain remained. That led to a loss of the ability to work for the patient and to walk without help. Multiple infection followed, which led to repeated hospitalization and many years of suffering. So this is actually a characteristic case report which could have occurred in Cologne in Germany or also in the United States here. This initially small ulcus then has destroyed more or less in a short period of time the well-balanced life of our patients. This demonstrates, I think, two things. First, that we have to really find an initial therapy, successful therapy of these small ulcers, and second, that we have to look for better therapeutic approaches in the next futures, and that's something where I would like to focus my discussion on today. So, chronic wounds are not only a severe burden to the patients, leading to the inability and to manage their life alone, it's also a challenge for the whole society. 18% of the patients, when you go to the literature, require hospitalization. In the US, there's a high individual cost which amount to a number of billions of US dollars. And when you calculate the loss of working days, you have about 2 million working days per year in the United States, which means that the overall cost of those chronic wounds is about 1 to 3% of the total healthcare costs. So it's significantly. And what the main information probably is, the main threat is that it does involve a number of the population today, but it's increasing with age, which puts the whole thing to a completely different dimension. It does put it to a different dimension because it's well known to you, probably better than to me even, that we already today pay a lot of the annual healthcare cost to age-associated diseases. And when you look to this figure here with the prognosis going to 2050, you see that the percentage, so there's a big demographic change in all Western populations, and that the projected proportion of people older than 65 years which develop aging-associated diseases really is increasing over time, which means on one hand that we have to spend much more of the national budget for aging-associated diseases in the next future, and it also means that we have to look for new developments which help us to decrease this amount of funding which we have to spend. And I think understanding the molecular basis of chronic wounds is one way really to address it. So for a biologist, looking to tissue regeneration and repair during lifetime is actually a very interesting problem, because wounds which occur before birth in the fetus or the embryo, they regenerate completely and you get a scarless repair. Then later in life, in young adults, scar formation is quite rapid, wound healing is rapid, but sometimes you get hypertrophic scars and keloids. And only later in life then chronic wounds develop because wound healing is disturbed. So this age dependency of tissue regeneration actually offers also opportunities. Because if you understand what's the molecular basis to get scarless repair in the before birth, then you could use those mechanisms and try to help and to modify scar formation in the young adult and in the elderly. So what do we know actually about the molecular and cellular mechanisms in wound healing? And I've tried to make a long story easy and short. And after the injury, a number of different steps occur with the invasion of many inflammatory cells from the circulation that then leads to the production of new vessels and new extracellular matrix. Wounds are contracted and then they are covered by the epithelial cells. What is important to note is that by that a storm of cytokines and growth factors are released, which more or less control with the orchestra the whole wound healing procedure. Changes in modification of those pattern in cytokines and growth factors will allow us to more or less modulate the whole process. So how do we do that? How can we really identify therapeutic targets in this complicated system? And this has been more or less started the story when Sabine Eming in our department has systematically collected exudates, wound fluids, which are easy to collect because you have just to cover the wound, then the wound is filling with an exudate, and then you can collect this exudate and you can analyze that. And what Sabine Eming has done is she collected exudate from nicely healing wounds and from non-healing wounds. And she started a collaboration with Jay Fox from Virginia and has made a proteome analysis and differentiate both healing and non-healing proteins in the wound fluid. And what comes out from this study is that in the healing wound, you do have a lot of extracellular matrix proteins accumulated, and you have a lot of inhibitors of proteases. In contrast, in the non-healing wound, you do have not inhibitors of proteases, but you do have a lot of proteases and proteinases itself synthesized and activated, which destroy, for example, cytokines and which destroy extracellular matrix proteins. And you do have so-called pro-inflammatory cytokines which induce an inflammatory reaction. So when she went to the literature and compared the pattern which she found in the non-healing wound with the data existing in literature, then she found a paper by Judith Campisi, who was interested in aging fibroblasts, in aging cells. And Judith Campisi has characterized those aging fibroblasts and found the secretion of proteases and inflammatory cytokines in these aging cells. And certainly now the idea came up, maybe in the aging population, wounds are not healing because you do have a certain pattern of aging-associated cytokines and proteases synthesized by those aging fibroblasts. And fortunately, there's one nice characteristic of aging cells, and that's the activity of so-called beta-galactosidase activity. And there's a paper which has already been published some years ago, where the group has tried to grow fibroblasts from venous ulcus and then looked to this beta-glucosaminidase activity, and they found high expression of this enzyme, which means actually we have some characteristic in the fibroblasts in those chronic wounds which are characteristic of aging senescent fibroblasts. And that then brought Sabine to this hypothesis that in normal skin after injury there are some cytokines released, for example vascular endothelial growth factor, which is inducing the growth of new vessels, which you need for new tissue production, and for example also transforming growth factor beta, which is turning on matrix synthesis in a number of cells. However, when you have old skin, you have an injury, then those senescent cells here, they induce proteases and pro-inflammatory cytokines. They make a hostile environment in the wound which is not healing anymore. So what kind of consequences does this have, for example, production of proteases? And Sabine Eming has then asked the question, why actually you don't have new vascularization? Why don't you have not new blood vessels formed in such a chronic wound? And I told you that the cytokine, the growth factor responsible for new blood formation is the vascular endothelial growth factor. And therefore, Sabine thought it's easy. I looked to the RNA for vascular endothelial growth factor in those chronic wounds, and she was very surprised to see that there was a high expression of indicating a lot of synthesis of those of RNA and also protein of this vascular endothelial growth factor. However, when she then looked for the protein present, she realized she could not detect the protein. So the only explanation then was that probably the protein is produced but is rapidly degraded by the protease present in those chronic wounds. And she could prove that by collecting exudate wound fluids from chronic, non-healing and healing wounds. And then she incubated an artificially produced vascular endothelial growth factor in those exudates, and you can see here that only in exudates from non-healing wounds there was a rapid degradation of vascular endothelial growth factor, probably by the proteases. So Sabine could then identify the protein which is mainly responsible for this degradation of this VEGF. She could identify plasmin as a responsible proteases, and then she asked, maybe we can use that now as a therapeutic target. There are two ways to do that. One way is, for example, to just block the activity of plasmin and other proteases, and the other way is to produce a growth factor, VEGF, which is resistant against the cleavage of plasmin. And Sabine has first started to really produce a mutant VEGF, which kept the biological activity, but she removed the cleavage site, the plasmin cleavage sites from this growth factor, and had now a mutant VEGF biologically active, which could not be cleaved by the protease anymore. She went to a mouse model, diabetic mouse with a chronic non-healing wound, and then added this VEGF into this chronic wound, and she actually could see an accelerated wound healing. So this was a nice proof that you can really design a therapeutic target and then use it in an in vivo model. However, when you go with VEGF, a mutant VEGF, to a human situation, then by systemic administration you have to be extremely careful. And therefore, Sabine went to Lausanne and started a cooperation with Jeff Hubble at the Technical University there. And what Jeff was a specialist was, he could bind growth factors to a fibrin matrix, and by that have a local, develop a local application of those growth factors bound to this matrix, and together they could really develop an angiogenesis-inducing bioresponsive scaffold, and then a patent was filed. This patent was sold by the University of Cologne to a company, and the company is now starting a clinical trial with that. Hopefully it will work really in the real situation. Let me go back to the initial discovery and again shows this picture from the proteomics identification of different proteins present in healing wounds and non-healing wound fluids. And I told you that in the healing wound fluids there is a number of proteins present which inhibit proteases. And I would like to point out a certain serpin peptidase inhibitor which is very highly upregulated in the healing wounds. And when Sabine now asks the question, how is this so-called alpha-1 anti-chemotrypsin regulated or present in the exudates or non-healing wound, shows that he found an enormous decrease of this activity in non-healing wounds, clearly showing that there must be a defect in that. So not enough in protease inhibitors in the non-healing wounds. So when he asked what the serine protease is doing, which can be inhibited by ACT, then the serine protease can activate many other proteases, for example, matrix metalloproteinase to degrade extracellular matrix, and it can also degrade certain cytokines. And what ACT can do is blocking the activity of proteases. And again, Sabine Eming produced recombinant alpha-1 ACT, added this ACT then to a non-healing wound in a mouse model, and as you see here, no granulation tissue, no scar formation here, but a lot of new granulation tissue and scar formation, closing of the wound in the wound which has been treated topically by alpha-1 ACT, which clearly demonstrates to you that by adding protease inhibitors, you can really enhance healing in chronic wounds. So you have two therapeutic principles. In chronic wounds, you have an induction of proteases. You could use protease inhibitors to inhibit the activity of protease, and you could also add mutant protease-resistant growth factors to overcome the activity of those proteases. However, I also told you that it's the inflammation which is turning on protease activity in these chronic wounds. And certainly now you might come up with the question, why not inhibiting inflammation at the first time? So inhibiting complete inflammation you cannot really do, because inflammation to a certain extent is required for normal wound healing. So you have to go a more specific way. And we can ask the question, can we specifically modulate inflammation? And when you want to specifically modulate inflammation, you have to target a certain cell, and you should use as a target a certain cell which has probably most influence on a number of other cell types present in an inflamed situation. And Sabine Eming concentrated on the macrophages as a major regulator of inflammation. And this macrophage here, it can really play an important role during normal and impaired wound healing, responsible being for phagocytosis, angiogenesis, and also for controlling cell migration and differentiation. So you can also look about the importance of macrophages for wound healing when you look during which period of the wound healing from day 1 to day 14 macrophages are present, and they are present more or less during the whole period of wound healing. And because we wanted to really dissect the mechanism which macrophages have play in wound healing, and therefore Sabine Eming has developed in a system where she, in the mouse, she can deplete macrophages specifically during a certain period of days. And when she did that and applied that and depleted macrophages and macrophage activity, therefore, also during day 5 to day 10, then she suddenly observed that those wounds do not close anymore. They have a model for a chronic wound healing situation. On the other hand, when she depleted macrophages in the early days of wound healing, then she got a completely different picture. Then she got a nice closing of the wound, but you could not see the scar at all. So it was something like reduction of scar formation, going to a more regenerative process and less development of scar tissue. So what it tells you is that you really can specifically interfere with the inflammatory reaction during wound healing, and we have to see actually whether we can translate that also to a human situation. So when you summarize the whole thing and ask the question, how is treatment therapy of chronic ulcerations today? Then we have a quite rough treatment by mechanical removal of the complete wound exudate. Also removal of all the nice growth factors which we might need. We do have a debridement, surgery debridement, and we use a number of different external dressings. Most of them do not work, or we don't know why they work, if they work. So what is tomorrow? I think tomorrow we can specifically inhibit certain proteases, and we could also use protease-resistant growth factors to overcome the hostile environment in a wound. And we can try to specifically modulate the inflammatory response. So, but is there something new in the future? And we have to keep in mind that sometimes in research, new developments come from completely different directions. And when you go through the literature and follow the literature, then you might know that there's an evolutionary conserved life-extending intervention by a number of measures, especially by caloric restriction. And that has been shown by in C. elegans, in Drosophila, and also the mouse. When those animals are not fed regularly, then life is extended considerably. And a number of groups have then also shown that caloric restriction not only leads to extension of life, but it leads also to a decrease of aging-associated diseases, including skin aging and hopefully also including the development of chronic ulceration. And we have to see whether this system also might lead to something which we might call hazy aging, with a complete reduction of aging-associated diseases. You have also to know that certainly many signaling pathways are involved in this process, which however converge to a relatively limited number of key steps. And certainly it might be a way in the future to modulate not only individual age-associated diseases, but by targeting the general pathways, then actually target a whole group of different aging-associated diseases, and hopefully then also skin aging and ulcerations. That works really in the mouse model, as seen in this mouse model, which have been published already 3 years ago, where skin ulceration have completely disappeared if you inhibit the RS1 signaling pathway. I thank you very much for your attention.
So are there questions for Professor Krieg? Yes, sir.
Hello. I was wondering, are you currently producing the mutant-resistant protease growth factors?
Okay, we have that, we can produce it, but we don't produce it certainly on a clinical scale. It's still experimentally.
And is that a small molecule or a biologic?
No, it's a VEGF, a complete VEGF with a mutation which is removing the plasmin-sensitive site.
Thank you.
If I could ask the questioners to identify themselves, that you might help us communicate a little bit better. Who had their hand up? Yes, sir.
Manager Phillips, City University of New York. Did you try synthetic inhibitors of plasmin? That's one question. And two, the matrix metalloproteases were present. Did inhibiting those also have a positive effect?
In previous days, many people have tried to use matrix metalloproteinase inhibitors to, for example, also in targeting tumor invasion. And they all failed. At least to my knowledge, they failed. And I think they failed because those enzymes have a variety of different activities. And some of those activities you really need for normal wound healing.
And the synthetic inhibitors?
We have not tried that.
Yes.
Do you know what the target is of the alpha-1 antitrypsin, alpha-1 anti-chymotrypsin?
It's a serine protease that I know, but not more specifically.
Probably not an arginine or
No, I don't know.
Any other questions for Professor Krieg? If not, Thomas, thank you very much. It's a great pleasure for me to introduce one of my colleagues and fellow faculty members at Columbia, Professor Angela Cristiano. Angela is the Richard and Mildred Rodebeck Professor of Dermatology and Professor of Genetics and Development at Columbia, and has had a remarkable career progressing from studies in inherited blistering diseases and understanding the molecular basis for these disorders, and then having moved into an understanding and a focus on the regulation of hair growth. And I'm sure she will share with you the fact that she's had a very personal interest in the problem of hair growth and the regulation of hair growth. And she has been a major force at Columbia along the lines that Professor Krieg was mentioning about when you're faculty members in relatively small departments that don't have large critical mass. The way you succeed in very large institutions is trying to leverage your strengths off of your base and reach out to larger entities and develop collaborations and develop meaningful interactions. And Angela has been singularly successful at Columbia in reaching out beyond our own department and building collaborative bridges in many different disciplines. And in particular, she's more recently been focusing on alopecia areata, which is an autoimmune disorder, and as we're now learning, a disorder that may be seen in association with other autoimmune diseases, and from a search for the genetic basis of these kinds of processes, we're beginning to learn that there are shared genetic bases that may well portend new thinking about how we're going to treat and maybe even hopefully prevent these kinds of problems. So Angela, it's nice to introduce you and look forward to hearing your remarks.
Thank you very much for this wonderful opportunity and for all of you for coming out tonight. I was just reminded looking at this initial slide, I think the first time I showed this was actually in Cologne in October, where I had the opportunity to be one of the first 5 or so Women in Science lectures. That was, it's become a very special invitation for people in the U.S. to go over and give this lecture. And I think that was actually the first time I showed this, so it's appropriate to show it again here tonight. So I'd like to just take a few minutes to tell you about some work we've been doing in the lab. Most of this is not published, so it's really very new. But I think there's a certain buzz, if you will, and you'll see why that's important during the talk, about this research, because alopecia areata is a skin and hair disease that's really been, for a long time, not very well understood. And so I hope that tonight, by the end of this, I can give you a flavor for why this has become, I think, an exciting area of research in skin. So alopecia areata is very common. Probably safe to say that, in addition to myself, there may be other people in the room affected. This is me, so I have the disease. So for me, this is not just business, it's very personal. But it's extraordinarily common. So many people have just a circle of hair missing on their head or a circle of hair in their beard that disappears from time to time. It can start like this with just patchy disease, and it can progress to the complete form that we know as alopecia universalis, where all the body hair is lost, and there are many different flavors in between. In addition, there are some clues that emerge from the clinical literature and also from anecdotal reports from patients. One of the most important clues is that alopecia areata tends to only affect darkly pigmented hairs, and it tends to spare white hairs. So there's some things that we know from the way it comes to the clinic that may actually help us understand about the disease mechanism. In addition, there are some people that say, I suddenly lost all my hair overnight, or you've seen reports of what's called sudden whitening of the hair. That's not actually the hair turning white. What it is an acute onset of alopecia areata where all the dark hair falls out and the white hairs actually remain. So there are reports of this throughout history. Marie Antoinette, among others, who report this finding. But there are all sorts of presentations of the disease, and it's fair to say that any ride in the New York City subway or anywhere in Europe, I think it's safe to say that this is extraordinarily common. Despite the commonality of the disease, though, for a long time, all we had were these histological clues that led us to understand, at least at the surface, what was going on. And so alopecia areata is really very carefully characterized by this histology. This is called a swarm of bees histology, and when a pathologist sees this, it's really a classic finding for alopecia areata. The nature of these bees, the immune markers, has undergone some evolution. It's been reported to have different types of immune cells that invade the hair follicle, and so I hope by tonight I can give you a sense of some additional work we've done to bring some clarity to this. So I just want to start out by saying that working in a medical center like Columbia is extraordinary because I'm surrounded by experts like my two colleagues here, and they're the ones in the picture on your newsletter in front of you. So Raphael Kleins is a cellular immunologist at Columbia, and Julian Mackay-Wiggin is a dermatologist and also runs our clinical research unit. So it's great for basic scientists like myself to work out the genetics of these diseases, but it's incredibly powerful when you couple this with immunologists who know what these genes actually do, and with brave clinicians who, with the support of our department and our chairman, encourage us to actually move these findings directly into the clinic as quickly as possible. So I think it's important that I acknowledge right up front what I'll tell you about tonight is really the efforts of all three of these of us. So again, my interest dates back to me becoming affected with alopecia areata, which happened about 15 years ago, actually, when I joined Columbia in 1995. And despite, again, the fact that we were interested in studying it, it was not yet approachable as a genetic disease for a number of reasons that we'll talk about. But I learned back then that, to my surprise, really, alopecia areata is the most common autoimmune disease in the U.S., if not in the world. And its lifetime risk is actually higher than these other autoimmune diseases taken together, which is really an extraordinary number when you think of the number of people who combined have these different autoimmune diseases. It's also likely that it's underreported. Most of this epidemiology comes from coding, from patient visits, and we know that many people with alopecia areata don't seek treatment because there hasn't been much available. So this is probably an underestimate. And yet, despite, again, the high prevalence, research into the basis of the disease has really lagged behind. There's a paper in the literature called The Burden of Skin Diseases, which Dr. Vickers is actually the first author of. And it's shown in that paper that among all dermatologic diseases, hair loss and alopecia areata in particular, especially in children, carries among the highest burdens of disease for any skin and hair loss disorder. And yet, despite all this, there's a Cochrane report that actually shows there's no evidence-based treatment for the disease. Even though intralesional steroid injections are commonly used, there has been no proper clinical trial that's proven their efficacy. So coming at this from a geneticist's point of view, it was clear that there was an opportunity here to begin to understand the basis of this disease, starting with an unbiased approach to take apart the genetics. What we know about it, and this is sort of to put you in a frame of reference for thinking about what happens in the disease, we can think about something called loss of immune privilege. So the hair follicle, like several other body sites, including the anterior chamber of the eye, the placenta during pregnancy, enjoys this property of immune privilege. Basically, the hair follicle is cloaked in molecules that keep it from being recognized from the immune system under normal circumstances. And then something happens. We don't know what the something is yet, but either a combination of an upregulation of danger signals or a loss of protective molecules reveals antigens potentially in the hair follicle that expose it to the immune system and begin to attack. These are those bees that you saw in the histology coming in and attacking the hair follicle. We didn't really know much about some of these things. By expression studies, Ralph Paus and his colleagues have shown up and down regulation of several factors in the hair follicle, but to actually get at what's driving the disease genetically, not a lot was known. But therapeutically, you can see already there's a lot of opportunity here if we think about intervening in this disease, because we have several areas where we can introduce potential therapies. One might be here at the level of the immune cells, another might be here at the level of the end organ or the hair follicle. So similar to what Thomas was talking about the interaction between immune cells like macrophages and the skin, in this case we have a deleterious interaction of these immune cells attacking a hair follicle. So when we think about a therapy, we're trying to basically restore immune privilege or shift this back to an immune protected state and allow those hairs to regrow. So we're talking about not just the genetics of the hair follicle in this case, but also the genetics of these immune cells. So it's really a complex disease in many ways. So to put you sort of again, in the notion of systems biology or genome-wide association, we're no longer living in an era where we can just look at one gene at a time. Although many of us grew up studying genetics in that way, the world has changed vastly. And with the availability of genome-wide technologies, there was, again, great hope that these applications would lead to a better understanding of disease. But actually getting that to translate has been a harsh reality. And the reason is that it's not just enough anymore to understand the genes that contribute to these diseases, because their effects can be very small. But looking at the effects of those genes in light of epigenetics, gene expression changes, putting those things together in a systems level way to then lead to an understanding of disease has become a bioinformatics problem. So functional genomics is the name we give to this process of trying to untangle these processes, how they lead to disturbance in what's called cell regulatory logic by my colleague Andrea Califano, who gave me these slides, and then using this logic and how it's perturbed to think about creative ways of intervening in the processes. So what I'll talk about today is the functional genomics of alopecia areata, starting with our genetic studies and hopefully taking you through to talking about some clinical trials where we've actually begun to move forward with this. Just by frame of reference, the genetic work took us about 15 years, and to get from here into the clinic has taken us about 15 months. So it shows that with a proper understanding of the disease, and again, motivated colleagues to help understand what's going on, things can actually move very quickly once we've uncovered the exact targets to hit. So just a word about alopecia areata. When we think about attacking a new genetic disease, it's important that we actually think about whether in fact it's genetic, and that's not always clear for each genetic disease. But here we had some clues about alopecia areata that were very convincing. First of all, it's highly inherited among family members, so a person with alopecia areata has a greater than 10-fold risk of first-degree family relatives also being affected. We see high concordance in twins, so identical twins have about a 55% or so rate of being both affected. That's comparable to things like psoriasis. Diabetes is a bit higher. Type 1 diabetes about 70%. It's also highly prevalent. Again, this is close to 5 million people in the US, so that speaks to the fact that it's a complex genetic disease. It can, in fact, be present at birth, and when it's present early, it tends to be more severe, speaking to more of a genetic enrichment. And then the most compelling thing was that previous candidate gene association studies, where many people before us had taken individual genes that had been linked to other autoimmune diseases and tested whether they were affected in alopecia areata. And by taking that gene-by-gene approach, many colleagues around the world have actually shown those associations. But by definition, a candidate gene study is biased because you're starting with a presupposition about the role of this gene in a given disease. And so what we wanted to do then was start with an unbiased approach and say, can we look across the whole genome and basically let the genome speak to us and tell us where we should be looking, not imposing any previous bias. So to do this, we use an approach called a Genomewide association study, which is basically a large case control effort where we're comparing individuals affected with alopecia areata to individuals without. These are unrelated, unaffected people, so basically complete case control design. Thanks to NIAMS, National Institutes of Arthritis, Musculoskeletal, and Skin Diseases, we were fortunate that NIAMS founded a patient registry for alopecia areata about 10 years ago, which defined this beautifully phenotyped cohort. This was just the first 1,000 patients or so, and so we've now got another 2,000 that we can add to this study. So we simply here compared 1,000 patients to about 3,000 controls using an Illumina chip, which just basically compares DNA markers across the entire genome. And we look for regions in the genome where certain SNPs or alleles are over or underrepresented in the cases versus the controls. So in the overwhelming number of SNP markers, these are equal, and so those are not statistically significant. But for very few, they actually are. So we see a skewing that tells us that a certain gene is overrepresented or associated with alopecia areata. And so when we do that, we come away with a diagram that looks like this. This is known as the Manhattan plot. It's not because we're here in Manhattan, but it's meant to look like the tops of buildings or smokestacks. These are individual chromosomes in the genome, 1 through 22. This red line is statistical significance, so 5 times 10 to the minus 7. Anything above that, as you see here, tells us that there's a gene of interest. And you can see these have p-values of 10 to the minus 35, so very significant. This large one on chromosome 6p is actually the HLA cluster, so a known susceptibility locus for many autoimmune diseases, but everything else is actually new for alopecia areata. And so this was very exciting that even in our first 1,000 cases, we were beginning to see a signal. Just by comparison, for other diseases, Crohn's, for example, type 2 diabetes, these studies are now up to 20, 30,000 patients, and still the genetics are not complete. So for us to see anything in 1,000 patients was very rewarding. I should also say that this cohort, again by NIAMS, was collected at 5 different centers in the U.S., Houston, Minneapolis, San Francisco, and Denver. Again, a 5-part registry that has now been converted into a clinical trials network where we hope we can use the same cohort of patients to start testing therapies. So what did we find? This is exciting because this was, again, the result of 15 years of hard work on many people's part. But there were surprises, many actually. So this is just a quick overview of the genes we found. Everything below this line represents the HLA cluster, so that large peak on chromosome 6, which we expected. But everything else above the line was actually new for alopecia areata. So the genes fall, interestingly, into 3 large groups. The green ones are involved in regulatory T cells. These are the beneficial T cells, the ones that keep immune tolerance in check. The red ones are actually interesting because they are ligands of a receptor on T cells called NKG2D. This is a bad one. These are danger signals. And so NKG2D is a killer receptor that's present on T cells that are going to come in and do damage to the end organ. And interestingly for us, the blue ones are genes that are expressed in the hair follicle. So for a long time, it was even questioned whether there would be a role for genes in the hair follicle at all in this disease. And so it was very nice to see some of the genes coming up expressed in the hair follicle itself. This one in particular is interesting, ULBP3 and 6, because it's both an attractant for those immune cells and it's present in the hair follicle, suggesting that this one could actually be very interesting. And we'll come back to that. So very quickly, we wanted to look and see what do we look the most like. And I think for alopecia areata, we were suspicious, as were many people, that we would resemble the genes for psoriasis and for vitiligo. These are the 2 most other common skin autoimmune diseases for which genes had already been established. We were very, very surprised to find actually that we have very little overlap with either one of these diseases. So excluding the HLA, which again was expected, we really found only 1 gene in common with these other 2 diseases. This was a big surprise because for years, any of the clinical trials that had been done in alopecia areata were done using drugs that were successful in psoriasis. So the assumption had always been that this was the right pathway to look at. And in fact, you see here, there's not a lot of evidence to support that. But what jumped out at us actually were some surprises, and that would be, what do we look the most like? And so just scrolling down this list quickly, you can see that we have an extraordinary number of genes in common with type 1 diabetes, with rheumatoid arthritis, and with celiac disease. And I think it's fair to say that most dermatologists would have looked at this and said, there's no way that alopecia areata has anything to do with these other disorders. What could possibly be in common? So this is about as big of a surprise as we could expect from this. We began looking through the literature, though, and it was clear immediately that the other three diseases, type 1 diabetes, rheumatoid arthritis, and celiac, had already recognized that they belonged to sort of a cluster of diseases. And so we've become sort of a fourth member of this group, looking again for evidence of what's called a common cause. Is there a common core of autoimmune genes that then predispose to susceptibility to a different end organ, depending on what genes and what damage is done either in the gut, in the joint, or in the pancreas? What's been very interesting, though, is that in all of these diseases, they've been frustrated by their inability to study their target organ. And so they've actually been very nice to us and very welcoming, because they're interested in the fact that we can biopsy a hair follicle and actually look at some of these immune processes in the end organ as the disease is going on. So this notion of common cause is something that we've begun to pursue. So in the rest of the few minutes, I'd just like to lead you through how we've begun to work from these gene targets to thinking about how to target this disease using both existing drugs, potentially ones that are in clinical trials, and then in the future thinking about how to treat it not just systemically by infusion or subcutaneous injection, but which would be most preferable for patients would be topical inhibition. So CTLA4 was the obvious first choice. This is the overlap of the genes between alopecia areata and the other three diseases at this locus, and you can see extraordinary evidence for association at CTLA4. Importantly, abatacept is actually a drug on the market for rheumatoid arthritis that's basically a decoy. So abatacept looks like this. So this is an antigen-presenting cell signaling to a T cell. This is signal 1. Signal 2 involves the binding of this protein, CD86, to this protein. And so what happens is, to prevent this activation, a soluble form of CTLA4 can come in here and basically act as a decoy. So this drug will swamp out the ability of this molecule to bind here and basically prevent those T cells from getting their marching orders, so they can't become killer bees in a sense. This generic way of suppressing the activation of these cells, though, has become a target not just for rheumatoid arthritis, but this drug is now being tested in a vast number of autoimmune diseases, over 75 or so clinical trials ongoing for abatacept. And you can see here, even though the drug was first tested in psoriasis, it's now, again, a massive number of trials using this drug because it's a general mechanism. So this is just a snapshot to show how it works in psoriasis. This was done by Jim Kruker over at the Rockefeller. This was before the approval for rheumatoid arthritis. It was actually first tested in psoriasis. So you see a very nice response in psoriasis patients taking this drug, showing that it is, in fact, effective in a skin disease, despite the fact that CTLA4 is not a candidate gene in psoriasis. So our question really was, could we extend that to alopecia areata and test that? So in order to do that, we needed to think about testing it in a mouse first. So there is a mouse model for alopecia areata that's called a C3H-HEJ model. So this is a grafted model where we take skin from an affected mouse and we graft that onto recipient C3H, so the same strain of mouse. We basically transfer alopecia areata with the presence of affected skin. If we just waited for the spontaneous mice to lose their hair, that would take about 9 months, and only 20% of the mice would do that, so it's not a great model. So to speed up that process and make it reproducible, we use this graft model to transfer affected skin onto C3H mice and watch them lose their hair in about 8 to 10 weeks. This is what the model looks like. So this is the graft site here, and eventually the mice will lose hair throughout. First on the ventral surface and then on the back. This model was developed by John Sundberg at the Jackson Labs and has been really a terrific model to work with. But most interestingly, I think this is very telling. Long before we came along with the suggestion of CTLA4, John Sundberg and his colleagues Joseph Carroll at the Jackson Labs actually tested mouse CTLA4 Ig in this mouse model, again in 2002, so 10 years before the genes came along, and actually showed the drug is effective in preventing disease in essentially all of their mice. They again used it because it was a generic mechanism and didn't know the connection to the disease at the time. But importantly for us, their study essentially provided off-the-shelf preclinical data that helped us convince the FDA that it was appropriate to use this drug in alopecia areata. And we're very fortunate that Bristol Myers has actually agreed to fund a small clinical trial for us starting soon in alopecia areata. And for this, I think this speaks to the ability of being able to do translational research. It's impossible to think of going from those genetic findings to a clinical trial in such a short time without incredible support. So the trial design looks like this. It's very standard. It's 6 months of once-a-week subcutaneous injection, and then a follow-up period for another 6 months to look again at the regrowth of hair, but also to do immunomonitoring. And that's to say, can we look beneath the surface of the skin to see what's going on at the level of the immune cells and actually track what's happening underneath the skin. So this gives you a flavor of what we'll be doing. So in addition to just giving drug and watching the hair grow back, we'll also be monitoring the blood, the immune cells, and the genome, gene expression, to actually follow what's happening as the drug is being given. And so these preclinical studies are generously supported by a grant from the Locks of Love Foundation. That's what's, again, in your newsletter right up there. And we're incredibly grateful for the chance to be able to do this work with these drugs. So that one's already in the clinic. In the last few minutes, I'll tell you just about what's next. And I think this is even more revealing about what can happen when you combine genetics with immunology. And so I'll talk now just for a few minutes about a new target. This one's called IL-15, and how we might think about targeting this one. So going back to those clues from the genetics, we reasoned that there must be something else that's in common between alopecia areata and the other 3 diseases that's linking us to them mechanistically. And so in the literature, we found evidence that in all 3 diseases, there's an upregulation in the end organ of one of those NKG2D ligands, one of those danger signals. So in RA, it happens in the joint, in celiac, in the gut, and in type 1 diabetes, at least in mice, in the islet cells. So the question we wanted to ask was, do we see the same thing in alopecia areata hair follicles? So this, again, is our second most significant gene on our list. This is ULBP3, the NK ligand. And we wanted to know if we saw overexpression of it in alopecia areata. This is going to be hard to see unless we can dim the lights a bit. Thank you. So this is what it looks like in the hair follicle of a normal individual. The blue is just the outline of cells of the hair follicle, and the red is staining for this protein. We see low levels of it here at the bottom of the hair follicle, but nothing really elsewhere. In patients with active alopecia areata, though, this is lesional skin where these hair follicles are under attack, you see a dramatic upregulation of this danger signal in the dermal sheath layer. This is the outermost layer of the hair follicle, exactly where you would hope it might be if you were going to assume it was an attractant, a ligand, if you will, for those killer T cells coming in. So strong evidence for upregulation here. We wanted to see if we could find the partner on the infiltrate. So looking at the cells attacking that hair follicle, we found both CD8 and NKG2D. So again, this is the receptor that binds to this ligand now present on the hair follicle. So as a geneticist, this was about as far as I could get on my own. And so I took this picture across the street to the lab of Raphael Klein's about 2 years ago now, almost. And he had worked on type 1 diabetes in mice, and showed him this picture, and fair to say that the rest is history. He's now jointly appointed in our department. You see his picture there in front of you, and he works at least more than half of his time on this problem. So it's been, again, an incredible gift to have someone just next door who's so skilled in immunology and could bring new insights to this. So I'm very excited by these slides. It's the first time I've shown FACS plots in a lecture. So what we're showing here is just a way of looking at, this is now in the mouse, skin and lymph node, showing you the presence of those killer cells. So just follow the circles here. In an affected animal that you see here, you see lots of blue dots in the circle. That's evidence of the immune cells. In an unaffected mouse, you don't see them here. here's a lymph node from the same animal. So in the alopecic setting, with hair loss, you see lots of blue and green here. Without it, you see far less. And we'll be looking at these as we show treatment. So this just helps show the identity of those killer bees, if you will, in both the skin, the lymph node, and spleen. So Raphael immediately made the connection to say, well, if that's the phenotype of these cells, and we know that they have this receptor, this killer receptor NKG2D, it's widely known in the literature that the fuel for those cells, what they need to stay alive, is a protein called IL-15. This is known to be an important maintenance factor for these cells. So we asked then, is this a potential therapeutic target for cutting off the oxygen of these cells? And so this is now moving to a human hair follicle. So this is IL-15 staining in human hair from a healthy individual. You see no staining for IL-15 or its receptor alpha. This is a chaperone that helps present IL-15 to the outside of the cell. But in alopecia areata patients, again, this is from active lesional skin, you see a massive upregulation of IL-15. This is a different layer now. This is called the outer root sheath of the hair follicle. And you see nice overlapping staining in the same cells here, suggesting, again, a role previously unknown for IL-15. This is now a cross-section looking at that same hair follicle in the other dimension, and you can see that those killer bees actually are armed, this time with IL-15 receptor beta. That's the receiving cell for the sending signal coming from the hair follicle, suggesting, again, a lock and key relationship between the immune cells and what's being expressed on the hair follicle. We then asked if that was reflected in the mice to see, again, could we use these mice as a model? So this is a normal mouse of a different strain called Black 6. You see no expression. This is a C3H mouse, an alopecia mouse without disease. And this is after grafting. This is a non-lesional site. On the mouse, you see the beginnings of expression. But in the lesional sites, where the mouse has lost its hair, you see massive upregulation, again, of IL-15, suggesting, again, an important role for the sustaining of this signal in the disease process. So turning again to what's going on in the pharmaceutical world, Amgen has two previous trials, both in rheumatoid arthritis and in psoriasis, of blocking IL-15. And because of the connection with celiac disease, this drug has recently been revived, and these studies are actually now ongoing. We, of course, then wanted to put the mice on IL-15 blockade right away, and so this just shows in two different sets of mice, so PBS, or saline controls, you see hair loss here on the belly after 9 weeks of grafting, but in the treated mice, you see no real evidence for hair loss here. This is, again, reflected at the molecular level, so sham surgery, we see low levels of this immune signature. After surgery, when the hair loss is induced, we see these genes going up, and then after treatment with IL-15, we see these genes going back down. And again, this is just a panel, a signature, if you will, of immune genes that are taken from an interferon gamma signature here. This is the work of Ali Jabari, who's a new faculty member in our department coming to us from NYU, and this has been his work and will continue. So the question is, have we gotten rid of the bees? So here you see bees that are present in the PBS-treated mice compared to the IL-15 treated mice. More of them here, less of them here after treatment, and that's reflected again here as well. This is a later time point now, 3 months in. You can see that 100% of the mice who were treated have kept their hair, as you see here, and those who were treated with only PBS, all of them have lost their hair. And here you see that their lymph nodes are of normal size, as is their spleen compared to the ones who were not treated. So we're very excited about this, and most importantly, you can see the beginnings of restoration of that immune privilege going on in the hair follicle. Just roughly, you can see these hair follicles are very red with staining, meaning that a lot of these molecules, immune molecules, are upregulated. And then here, after treatment, you see those signals going back down, suggesting that we're moving them towards a more immune-privileged phenotype. And again, here you see from the skin the presence of those bees in untreated mice and the absence of those bees after IL-15 treatment, suggesting that we're targeting the right cells. This is the early time point, and this is now week 13, showing that those genes are, in fact, continuing to be repressed. So I won't dwell on this, just to say that we'd like now to move downstream of IL-15 and see if we can do this better and do it topically, again, making it more acceptable for patients. And so our next frontier is this class of kinase inhibitors. particularly targeting the JAK-STAT pathway downstream of IL-15, and importantly, thinking about ways to attack, in addition to the hair follicle, those immune cells as well, again, coming at this from both angles. So I hope I've convinced you about some of the power of using these techniques in a disease like alopecia areata, where we're dealing with essentially two dysregulated organ systems, trying to untangle that genetically and essentially rebuild that system in a way that we can fix a broken immune system, which is not an easy task, as I've learned. But again, with the help of these wonderful colleagues, I'm delighted to say we're a little bit closer. This is just a closing slide from Francis Collins, who's the director of the NIH here in the States. Francis was one of the earliest proponents of using this method of genome-wide association to identify new disease gene targets. And so this is a paper of Francis's where he showed some success stories. Total number of GWAS genes. Again, type 2 diabetes has 44 genes. Some of these have been doing this a lot longer than us. And here's how their genes have translated into drugs. And I'm delighted that we can now add ourselves to this table, even though we only have 8 genes. One of them so far happened to hit upon a marketed drug, abatacept. And so we're enormously pleased that this is moving forward into the clinic. And I'll just close by thanking my funding sources here, particularly NIAMS, the National Alopecia Areata Foundation, who helped us get started in all this, and most recently Locks for Love, and particularly our New York State funding bodies, the American Skin Association and the Derm Foundation, my colleagues at the AA Registry from those 4 sites, Vicky Kalabokis from NAF, who again works tirelessly with the patients to get this work done. And here my 2 colleagues just down the road from Cologne in Bonn, Germany, Regina Betz and Marcus Newton, who are working independently on alopecia areata. And we've now begun to combine our forces and do a meta-analysis of their genetics with our genetics in the hope of bringing together, again, a large cohort to make our genetic power even stronger. And once again, a thanks to Raphael Kleins for really being in this from the beginning. Thank you very much.
Thank you very much, Angela. Questions from the audience? Yes, sir.
Vanderbilt City University. At the beginning of your talk, you emphasized the effect of hair color on the disease. I was expecting you to come back to that at the end, but you did not.
I didn't. So buried in one of our slides, if I can go back, none of our genes were obviously connected to pigment, except one, which I didn't really mention very much, you're right. So people have looked long and hard for an autoantigen for alopecia areata, and the source of looking for that autoantigen has been the melanocyte. The reason is, the thinking is that the hair follicles are undergoing a bad apoptotic death during their hair cycle, and perhaps pigment antigens are being exposed. And so the reason I think we thought for a long time we'd be similar to vitiligo is because those same cells in the epidermis are the targets for vitiligo. We haven't been able to find any of those melanocyte antigens, but we did find this gene that I didn't mention, syntaxin 17, which is buried here. It's one of the hair follicle expressed ones here, and it's involved in premature hair graying, not in humans, but in horses. Lipizzan stallions have a mutation in syntaxin 17 that's the cause of their gray hair pigment. So it seems to be involved in melanosome transport. It's a snare protein. We haven't yet delved deeply into either this one or this one. Ironically, we're leaving the hair follicle genes for last. But this was the only hint about that pigment connection that emerged from the genetics. Sure.
Some other questions? Yes.
Is DHT a factor in alopecia areata?
So DHT refers to dihydrotestosterone, which is one of the metabolites of testosterone that's commonly associated with male pattern hair loss. So none of our genes that you see here have anything to do with androgen pathway. Using the same genetic approach, Marcus Newton from Bonn and his colleagues have shown an association of the androgen receptor with male pattern hair loss, not with alopecia areata. So it's clear that it plays a role at the level of male pattern hair loss, but not that we've seen yet in alopecia areata. Sure.
Must be some other.
There is. Everyone's hungry. They want to drink.
Yes.
I'm Stephanie Schumacher from the German Center for Research and Innovation.
Hi.
Obviously, I'm a layperson, so these common genes that are shared with psoriasis, for example, or for rheumatoid arthritis, are both of those diseases often commonly expressed in people that possess those genes?
It's a great question from a layperson or anybody. So the question you're asking is, do we see more than one autoimmune disease in these patients? So we call that comorbidity, which means can we look across disease groups? So we've done something, we've looked in the serum of alopecia areata patients who don't know whether they have celiac disease or type 1 diabetes. And about 10% of alopecia areata patients carry celiac antibodies as well, or type 1 diabetes antibodies. Interestingly, the other 3 diseases have done those same studies, and the rates of antibodies in their serum is about the same. It's between 8 and 10%. But psoriasis has a severe form that's called psoriatic arthritis, so there's clearly some comorbidities there. Many patients with alopecia areata will report anecdotally they go on a gluten-free diet. Some of them grow back their hair. We've now begun to raise awareness at Columbia about asking the questions about alopecia areata. So it's a very subtle way of asking the question. If you say, do you have other autoimmune diseases in your family? Most people won't think of alopecia areata, they'll say no. If we say, are you losing their hair? They'll say, oh, I have male pattern hair loss, but I don't have any. If you say, are you losing your hair in circles? Suddenly everybody's got alopecia areata. Their cousin, their child, their neighbor, everybody. So now that we're asking the question better, we happen to have at Columbia, believe it or not, a world-class type 1 diabetes center, 1 floor below us, a huge center for celiac disease that follows about 2,000 patients, and most recently a new division director in rheumatology whose focus is rheumatoid arthritis. So suddenly patients are coming to us with many of these comorbidities. But this is well established not just in our 4 diseases, but across all autoimmune diseases. And the thinking there again is that there may be this core of susceptibility genes for a broken immune system, and that what distinguishes them are changes in the end organ that target that broken immune system in one organ or another, which should make therapy even easier, because thinking about targeting common mechanisms is something that, again, you can see from the CTLA-4 work could be applicable across many of the diseases.
Yes.
Sure. Why do you think that it's focal, that you get circles?
That's a brilliant question, Dan. Thank you. There's an old answer and a new answer. So the old answer is, and I think it's now becoming clear from the work of Tom Kupper and others, is that many, I won't speak to the experts, many dermatologic conditions have a memory. So fixed drug eruption, many diseases that involve immune cells come back in the same area of the skin where they initially showed up. Tom Kupper's work now recently has defined resident immune populations in the skin that stay there for many, many years and actually don't traffic very much. And so Tom's hypothesis is that there's a memory, if you will, in the skin that actually drives some of these things to be focal. The reason for the circles is a newer answer, and there's no evidence for this except just speculation. But Cheng-Ming Chong and his colleagues at UCLA, working in the mouse, have recently shown that for a wave of hair cycling to progress through anagen, through the hair cycle, there has to be a change in the dermis. It has to become competent to allow a wave of hair growth to progress. And in humans, they cycle differently than mouse. So mouse hairs cycle largely in a wave, and then in large chunks of mouse skin cycle together. So you see islands of mouse skin that cycle at the same time. In humans, that's not exactly true. So human hairs cycle each one individually, which is why in the normal setting you don't see patches of hair loss when the hair cycle is asynchronous. In alopecia areata, the hairs become synchronized and they go into catagen, they go into regression in a uniform way. So one way of looking at it is that maybe you're hitting a spot of competent dermis. So what happens regionally is the same thing in Cheng-Ming's mice, that there has to be a change in the dermis, largely BMP driven, that allows this wave of hair loss to progress. So that maybe on human scalp there are in fact regions where competence is different, even though each hair is cycling under its own clock. It's not the answer you're looking for. I'm sorry that we don't have an easy explanation, but we are beginning to, I think about how to relate some of what's coming from the mouse work to what might be going on here.
I actually have a question, which is, we know that in some instances, alopecia areata will resolve spontaneously. What can we learn from that vis-a-vis this biology?
So, yeah, about 20% of patients will report spontaneous remission of disease. We have tried to stratify their genetics on the basis of whether they're spontaneous remitters or not. We don't see anything in the genetics that leads us to believe much about that. We haven't had an opportunity really to ask for patient samples from those who have undergone remission. So to see whether their ligands have gone back down spontaneously, whether their immune cells have resorbed or left the lesion. So my sense is that there's probably something to do with the restoration of the hair follicle, and I think that's just my hunch, is that perhaps spontaneous remission is a correction, a self-correction in the follicle, and not so much on the immune side. But we debate this hotly in the lab. But there's certainly a lesson there. This is unusual for autoimmune diseases too. Many of them flare and wax and wane, but not too many just go away on their own like that. So something we'd like to do is convince patients with who've regrown their hair to give us a biopsy. It's not easy.
Okay. Yes.
So, Professor Krieg and Professor Cristiano, so what extent are your postdocs and your graduate students actually going back and forth to the U.S. and Germany? You mentioned Bonn and you also have Cologne. Is there any exchange at that level?
Okay, that's just one of those strategies we have been thinking for the last few months, and especially also just today, that we establish an exchange system at PhD student level for a short period of time between our collaborative research center, which we have funded by the German Research Foundation, and by the program grant, which is funding here the Skin Research Center.
Maybe I could ask a question of the audience, and that is, how many of you have current collaborative or cooperative arrangements, those of you in educational institutions or elsewhere, with counterparts in Germany? And would you be willing to share with us what those are? So your mission to foster this seems to be a very good idea. Well, I can say that the opportunities for, as I'm sure most of you know, in today's world, the funding from the NIH, at least in this country, is extraordinarily competitive and extremely difficult for, particularly for young people, to launch their careers. And this is cause of growing concern in terms of the future of our biomedical research enterprise. Thomas mentioned earlier today when he was visiting the department that in Germany, the funding rate for the DFG, which is kind of the NIH equivalent, can be as high as 25 or 30%. Is that correct? Now, there are differences because philanthropy is not as prevalent in culture in countries outside the U.S. as it is here. But nonetheless, I think it's going to be important for all of us who are heavily invested in and concerned about the future of biomedical research that we, and particularly with physician scientists. Physician scientists today, again, I'm sure you realize, particularly in this country, when they finish their education, they typically have debts of several $100,000, and for them then to embark upon a research career where the compensation is not so generous presents many different challenges. So this opportunity for us to cooperate between Columbia and Cologne, we hope is a small step in which we can hopefully fertilize this kind of collaboration and hopefully perhaps in the future even seek opportunities for jointly funded research projects if that becomes feasible. So are there any other comments? Would the people from the German Center for Research and Innovation have any comments or points to make, you'd like to make?
Yes, I'm Joanne Halper from the German Center for Research and Innovation. First of all, I'd like to thank our speakers and our moderator for your excellent presentations. Thank you very much. Let's give them a hand. And I'd like to invite everybody to continue this discussion outside. Just walk out to the door on the left and we have a little networking reception for you. Thank you.